later it's commercialized as a probiotic, but this strain, by his nature, it's producing a combination of L and D lactic acid. Even if you open the Burgi is like a Bible for the microbiology because everything is well described and systematically explained. They say they're producing almost equal quantity of L and D form of lactic acid. So you're obviously not supposed to give this probiotic for babies. And do you know what? I found on the market drops for babies containing Lactobacillus reuteri. I mean, these people, they developed this product, they never opened the book of microbiology. What's happened? Because industry, sometimes they ignoring, they want to make a profit, and they're ignoring basic rules of safety. Sometimes I like, actually not some, always I like safety is priority. When we work with the lactic acid bacteria in the lab, first step is to check the safety. And we're going to have some slides about this, but safety like hemolytic activity, production of the lactic acid, we're checking for the antibiotic resistance. Because when we talk about the lactic acid bacteria, always we think about they're good guys. They are beneficial, they're good, not all of them. Okay, genus Streptococcus. Which one coming to the brain first? Which one? Thermophilus, right? Because of yogurt production, right? But then it's only 3 streptococci that are considered as a safe. Thermophilus, Macedonicus, actually Goliticus species Macedonicus, and Salivarius. The rest is pathogen. Streptococcus pneumonia, need to convince you that it's a pathogen. The word is already telling, related to pneumonia. So most of them could be very dangerous, some of lactic acid bacteria, not all of them. Enterococci. Enterococci resistant to vancomycin. It's a nightmare in the medical healthcare because they are enormous problem. So you see, it's not all of them are nice, but some of them are very good. But then we need to check for this. Then how we can apply them, these microorganisms? Okay, we can use them as a starter culture, obviously, or the fermented food. By the way, something interesting, India is a very rich of fermented food made by bacillus. Not by lactic acid bacteria, but the genus Bacillus. And several of these products was having very good health promoting properties. And some of these bacteria producing bacteriocins as well. They're producing antibiotics, they're producing surfactants. They're really extremely good metabolitic factory because they're producing several. They've already been explored, the bacillus in the pharmaceutical and chemistry and biosynthetic processes, but they are really a huge potential to make a new probiotics because remember, most of the bacillus, they're spore forming. Lactic acid bacteria are not, but bacillus is spore forming. So you can actually produce the spores, you can keep them for the longer, and you can apply them as a probiotic much more easily. And it's one advantage, especially in the animal industry, production of farming, they're using bacillus producing probiotics for the chicken, for the ruminants, they're using even for the human application. Bacillus coagulans, that is changed the name, now is Hendrexia coagulans. It's one very popular probiotic, not lactic acid bacteria, but bacillus. And it's having application in several and authorization in several countries. Even Way Santos, he ran away. He got bored with me and ran away. Okay, Santos, what we are here, it's another group of microorganisms that as well can produce bacteriocin, and some of them even can be considered as a probiotic. Especially for the agricultural applications, very often bacillus is much more applicable because lactic acid bacteria, they're difficult. They want a rich system to grow. Bacillus is much more simple and they can grow much more easy in the agricultural applications. So some of them of lactic acid, but well, we stick now to the lactic acid bacteria, they can be considered as a probiotic or postbiotic. And we're going to discuss these two terms a little bit more details later. Actually, I think on the 3rd or 4th day. Technological properties of this lactic acid bacteria in the food production, improving sensory characteristic, making products more pleasant, extending the shelf life. Why? Because they produce acid. Salmonella-like acid? No. Salmonella pH go below 4.2, 4.3, it's already not very happy. And lactic acid bacteria, especially formal lactobacillus genus, pH can drop to 3.7, 3.8. Salmonella is, well, you cannot kill the salmonella, but salmonella will not multiply. It's already something, because salmonella is a big problem. Then improving the color stability. Some of the metabolites produced by the lactic acid bacteria, they can have influencing indirectly the colors of the products, or even improving the texture of the food products. So let's see, how do you like yogurt? Thick or liquid here? Well, in Bulgaria as well, yogurt needs to be thick. In Brazil, they wanted liquid. I don't understand why. Reason, the lactic acid, Lactobacillus bulgaricus, responsible for fermentation of the yogurt, is not growing very well in Latin America. I don't know, must be nationalist, this strain, and don't want to grow there. So they actually adapted consumers to like it more liquid. Okay, it's not bad. But again, if you use a leuconostoc that produces hexapolysaccharides as a co-culture, you actually can improve the texture of your fermented dairy products, because hexapolysaccharides made it more thicker. Well, another important point, if we talk about yogurt, yogurt is only called yogurt when it's fermented by Lactobacillus delbrueckii subspecies bulgaricus, and in collaboration with Streptococcus thermophilus. All the rest is fermented milk. So this is important point. Again, very often people going in this error when they submit a paper, they ferment it with a different, even bifid or whatever, and they call it yogurt. Sorry, no, it's fermented milk. So it's a kind of terminology. But then bioactive compounds produced by this lactic acid bacteria could be variety and variety. You can have it acid, obviously it's lactic acid bacteria, so they will produce lactic acid. They can even produce different acids, depends if they're homo or heterofermentative. They can produce hydrogen peroxide, carbon dioxide, some of them. Heterofermentative, they do carbon dioxide. And this can give this fizzy, who like a kefir? Or no one like a kefir? Kefir have these bubbles because of production of carbon dioxide. Well, most of them is because of the yeast involved in the fermentation of the kefir, but some of them, some heterofermentative lactic acid bacteria can help in this production. Low molecular weight metabolites, several of them, like reutherine, it's not a bacteriocin, it's not an antibiotic, it's a reutherine. It's completely different structure, but they are there, they're produced by most of them. Actually, they say it's only Limosilactobacillus reuteri producing reutherine, but again, some Lactobacillus fermentum, that now is Limosilactobacillus fermentum, as well can produce it, reutherine. We have the diacetyl. Diacetyl is very important because diacetyl have the kind of antimicrobial properties. Even in the beginning of 20th century, they was using for treatment of tuberculosis, but you needed a lot to treat. So actually having some side effects, but again, it's in the beginning of the 20th century, there's antibiotics. No, the people don't have choice. They look for something that can inhibit the Mycobacterium tuberculosis. Several enzymes produced by the as a bioactive metabolites by the lactic acid bacteria. Of course, bacteriocin, and again, interestingly, proteinase inhibitors. Why I put them separately and not part? Well, they inhibit the growth of the microorganism. But very often, if you detect activity of bacteriocin against viruses, it's not a bacteriocin himself. It's these proteinase inhibitors, because in this way they're able to inhibit the growth of the virus. Actually, not the growth, multiplication of the virus, because the virus go, inject the DNA or RNA, whatever virus you have, and he needs to produce all these components inside the host. But since you have these proteinase inhibitors, they can inhibit some of the proteases involved in the reconstruction process of the virus. And what will happen? No virus. Well, we have some examples. We found some protease inhibitors and some bacteriocins that work against herpes virus, they work against measles, polio virus. And unfortunately, we tried against COVID, it was not working, so we don't have panacea. You cannot kill everything. But again, we need to try, we need to explore. By the way, what's the problem with polio in India? Polio virus. It's a problem because in Brazil it's starting to coming back. It's really starting to coming back. The problem is we have the vaccine, no problem. The problem, people don't want to do the vaccination. And if the polio is coming to the child, this child will be paralyzed for the rest of his life, but this will ruin the life of the family as well. It's a very social and medical problem, not just a simple problem. Well, something, again, very classical, very typical, but why it's important. This is the difference between gram-positive and gram-negative. But actually, this is very important when we're going to discuss mode of action of bacteriocins, because of this difference on the cell wall, bacteriocin actually from gram-positive can work against gram-positive, and bacteriocin from the gram-negative will work against gram-negative. And this is actually something that is a big challenge for the science, because if you develop the medical formulation, you want to kill all the pathogens. And you want to use it such as a more simple one, more safer one producer, but you want to kill more dangerous one. And the problem, most of the pathogens, they're gram-negative. So how are we going to kill them? Well, don't be so disappointed, because Staphylococcus aureus, it's a gram-positive, it's a really a big problem. If you think about Staphylococcus aureus can be involved in the osteomyelite, can be involved in the skin disease, can be involved in the food poisoning, can be involved in the meningitis, can be involved in the respiratory disease. So technically any part of our body could be attacked by Staphylococcus aureus. And the good point, bacteriocins from gram-positive, sometimes we need to select the appropriate one. They can kill gram-positive Staphylococcus aureus. But again, look the structure, look the formation of the cell wall. All these things actually having importance later when we're going to discuss the mode of action of the bacteriocins. And gram-positive, you have the peptidoglycans, quite simple structures. Well, down we have some proteins, polyphospholipids, etc. But again, on the gram-negative, it's much more complexity. But again, you have lipopolysaccharides, and this could be a good receptor. We have some proteins in the other membrane that can be a good receptor. And this is actually important for the mode of action of the bacteriocins. Okay, I have a question. You want me to try to do the 2-hour lectures in one block, or you want to have a break in the between? How do you prefer? It's a democracy. You can say, but then I will take a decision. Can we do it in one block? Yes. Okay, no problem. So just please let me know when it's approaching time to finish, because I will continue. Tomorrow we will continue when we stop today. We'll continue tomorrow on this stage. But again, you need to give me a sign when I need to stop for today. Okay? Deal? Okay. Some examples here with the morphology. You see, it's a kind of interesting microorganisms. Sometimes I say actually they're beautiful when you observe them in the microscope. But now it's coming some interesting points here. When we're talking about the fermentation process, it's actually transforming the carbohydrate to the energy. Fermentation is not something that microorganism want to do for us. He wants to win the energy. And we have endogenous electron receptors in the fermentation. In the respiration process, that is completely different thing, you have exogenous electron receptor. So it's very simple. Normally fermentation is an anaerobic condition, when there is no oxidative phosphorylation to maintain the production of ATP. This is a rule. In anaerobic condition, there will be no oxidative phosphorylation to production of ATP. During the fermentation, pyruvate normally is metabolized to the various compounds, and in homolactic fermentation, production is only going to pyruvate and then to lactic acid. It's rule. In a heterofermentive fermentation, you can have the different final products and different acids, and even alcohol that could be produced. Well, in the yeast it's a little bit a different story. But again, in the lactic acid bacteria, homofermentative process, it's going down to the lactic acid. In heterofermentative fermentation, you can have acids of different variety, but lactic acid will be for sure. There's no way to escape. So we have these pathways, very simple, already had this in the books of microbiology. The professor asked you to remember all these pathways. I don't want to remember this because it's more important. I mean, okay, we have glucose 6-phosphate, fructose 1,6-biphosphate, but you have glucose. You're going to glyceraldehyde 3-phosphate. Then going down to pyruvate, going down to lactic acid. Glucose, pyruvate, lactic acid. In heterofermentative, you can have the glucose, but you can have pentose as well. And very important, are you going to be university professors or you want to go to the industry? Who wants to go to the industry? Hands. Industry, give them money. They pay well. Who wants to be a professor in the academic? Well, you need some time to show the students heterofermentative in the lab. Normally take one leuconostoc. For sure leuconostoc will produce this gas, CO2. You even take the glass tube, you put on the vortex. You have a huge amount of foam. It's already showing the presence of CO2. If you have production of CO2, obviously you have the heterofermentative microorganism. And this will help you later in the taxonomy. Normally people are always complaining that it's too costly, too much work, this biochemical, physiological test, but we need them. Because very often when the people talk about the identification of lactic acid bacteria, everyone, okay, genetic. Yeah, genetic. Try to do the identification of enterococci by genetic methods only, not going to work. 16S sequencing is a very good tool. It's helped a lot. But the problem, when you do the sequencing of 16S in the enterococcus, try to blast the results. What you're going to have? 99.9% enterococcus fecalis, 99.9% Elococcus munti, 99.9% enterococcus caselinoflavus. Which one? The problem, the 16S segment for the enterococci is very conservative. It's not helping. You need to go back to biochemistry. You need to go back to the physiological test in order to have appropriate identification of enterococcus. Same thing with not lactic acid bacteria, with bacillus. And you're going to be scared. Soy milk is popular in India or not? Okay. Soy milk very often is contaminated with bacillus. The fact because it's a plant material. Plants go where? On the ground, no way, and the bacillus is very present in the ground. So you have contamination with bacillus, and when bacillus make spores, so you pasteurize the soy milk, you put in the Tetra Pak, you put on the market, and 3 months later you see the Tetra Pak look like a balloon. Well, bacillus grow, the spores survive the pasteurization, they grow. You do the test in the lab, and you came like a scary Bacillus anthraci. It's not, it's Bacillus subtilis most of the time. Well, it could be Bacillus cereals, that is not a good one, but very often it's Bacillus subtilis. Problem is the same thing. 16S in Bacillus giving a very same results for several Bacillus because it's a very conservative. So obviously, again, genetics is a good tool, 16S is a good tool, but it's not it's not a cherry of the cake. We need to know basic biochemistry, we need to know the physiology of this microorganism because it's a complex way to do the appropriate identification. And then, very often people making this error, they say the lactic acid bacteria grass, generally recognize it as safe. Well, more or less, but general, just a general, not a real. The problem is such a big group of microorganisms, lactic acid bacteria. As I mentioned to you, most of the streptococcus, they are lactic acid bacteria, but they are pathogens. And they are very dangerous pathogens. Half of enterococci, they are carrying vancomycin or methicillin resistance, and they are a problem. You don't want to go in the hospital and to catch vancomycin-resistant enterococcus. No way, because what antibiotic we're going to use now with you? Because vancomycin was considered like 10 years ago was a last option. Now we're having some new, but again, it's It's not easy. And then you'll be surprised, some of the species that are considered traditionally safe, like the Lactobacillus plantarum. It's considered traditionally as a safe microorganism. Some of them associated with the inflammation of the heart, human heart. Some of them can carry virulence genes. I have some examples later doing a kind of screening for virulence genes in the different lactic acid bacteria. And you can really surprise to see how much virulence genes could be present and can make this strain very nasty and a good candidate to receive the name of Humadin-Law. Well, another point that needs to be always taken into consideration, production of biogenic amines. Biogenic amines, it's, we're going to have more about this, but now very shortly. They are decarboxylation of the amino acids, normal physiological biochemical process in the transformation of the organic substances. The problem, some of these components actually could be toxic, and they can be influencing even the brain function, some of the biogenic amines. So if you have, let's see, non-fermented food products, and there was a biogenic amines production there, well, there may be a little bit, but the problem is when you have fermented food products. These fermented food products, especially probiotics, they need to be alive. They need to have a live microorganism. And they can produce all the time biogenic amines. So they can accumulate in the product. And in fact, in the time you can have it a product that is actually coming to the toxic levels, these biogenic amines. So this is another important point when you do the work with the lactic acid bacteria. We need to take care to don't select none of the microorganisms that could be potential problems. Well, if I develop new probiotic, I want to put something on the market that is safe because I want to have sleeping in the night conscious. I don't want to have nightmare that someone gets sick of my product. So, right. Let's be honest, and money is not top thing in the life. First of all, we need to be responsible, and we need to produce the things that will give the benefits, not negative points to the consumers. Now, lactic acid bacteria I already mentioned, but let's repeat again. They producing organic acid, diacetyl, hydrogen peroxide, carbon dioxide, bacteriocin, low molecular weight, and they was used these antimicrobial products produced by the lactic acid bacteria since the human civilization exists. All the fermented food products produced all around the world in all the civilization, they involved lactic acid bacteria, they produced these antimicrobials, they protected the product, they've been used by the traditional medicine and even with the modern medicine because these days probiotics with the bacteriocynogenic property and producing other antimicrobials, they are actually having more and more interest from the industry because they can have the better application. But coming to the question, what is the bacteriocins? I was using this term several times, and still now I was telling nothing about this. Well, definition that is accepted is like almost 30 years, even more now. It's saying this is the ribosomally reduced polypeptide. Well, why ribosomally important in this definition? Because you can have it antibiotics with the protein structure, but they are produced and then they have the enzymatic modification in the cytoplasm. In the case of the bacteriocin, they are produced in that way, and they work as a produced. They will have very slight modification after that, only with one specific groups. And normally they are active against closely related microorganisms. Meaning again, remember I was mentioning in the beginning, bacteriocins from gram-negative, they kill gram-negative. Bacteriocins from gram-positive, lactic acid bacteria, they will kill pathogens on spoilage of gram-positive. Well, this definition actually needs to change, and we're already suggesting some changes, because if you look the history of development of the bacteriocin, they start in 1925. 1 century. bacteriocin already on the scientific literature. First one actually was colicians, produced by Escherichia coli in 95. Then a little bit later we have bacteriocins from gram-positive. And then originally they was introduced as a biopreservation tools. Well, very good, so we can protect our food products. Well, our grandmother was protecting them because of fermentation and production, but now we know what is going there, why they work, this fermentation process, why they're protecting the food product. Well, later it was discovered that some of the bacteriocin from lactic acid bacteria, gram-positive, in fact can kill gram-negative. Well, what's going there? Why it's working? Why it's happening? They realize, in fact, very often it's not a simple interaction between bacteriocin and gram-negative, but there is a synergistic interaction between bacteriocins and some surfactants. Well, who work with the lactic acid bacteria again here? Using MRS media? Yeah, they work very well. What is in the MRS? Twin 80, surfactant. And twin 80 together with the bacteriocin actually working together in the synergistic effect. In this way, first twin part of the media actually will make a change in the surface of the gram-negative, and then the bacteriocin coming and do his job. In fact, it's not just a simple bacteriocin, but then we get a little bit further. We wanted to purify the bacteriocin because we want to go everything out of the surfactants. Well, we know if we want to kill in the pharmaceutical products, you can mix not only bacteriocin, you can put the surfactants, you can put everything that you want, you make a complex, because the pharmaceutical product is not a simple single molecule, it's a complex preparation that everything needs to interact and do the best way of the job. Stability, efficacy, etc. Well, we purified the bacteriocins because we had one produced by Enterococcus multi that was very good activity against gram-negative. So we purified, and we had a single peak on the HPLC. We do a mass spectrometry, confirming we have the pure product. Still work on the gram-negative. Well, the problem is we stop because the bacteriocin been taken as a patent from the medical pharmaceutical company, and they tell us that we're not allowed to publish any more results in mechanism of action, but they wanted to know what's going on. Well, it's very simple. We discovered that we are attacking different receptors in the gram-negative, and attacking this receptor, bacteriocin having the option to do his job even against this gram-negative. But if the gram-negative don't have this specific receptor, they cannot work.
So the patent was on the sequence of the
bacteriocin and application. Well, we're not allowed, I mean, I don't know, some countries they allow, but in most of the world, we are not allowed to patent microorganism because something that is from nature. We cannot patent something that has always been there. But we can patent application, we can patent the products produced by this microorganism. The patent also peptide and the killing these gram-negative pathogens. By the way, companies from India.
That must be a US patent?
No, international. Because the work was done in South Africa, together with this Indian company that had the branch in Australia, in South Africa. I think I'm not allowed because of the record to say the name because we'll be like advertising the company. But it's a company from here. If they pay me the royalties almost 20 years, it means they're selling the product. But they was very strict to don't release the mechanism of action. It's, well, I'm releasing, it's a different receptor for the bacteriocin that it's managed to attach to this receptor, but I'm not giving structure of receptor. It's helping something. Well, but I don't see the problem with this. Well, anyway, later some of the effect of the bacteriocins was shown to work even against Mycobacterium. Now, okay, I will do something. This doesn't matter. Okay, let's think about something. Now we're in the contact like 1.5 meter. If I'm having tuberculosis, she's already in trouble. 1.5, 2 meters, it's enough social distance to transmit. Hello. Congratulations, you have it. Okay, no, don't worry. I need to do test every year because of the safety issues and work in the lab for the TB. But the TB, it's a big problem because you can catch it in the social contact. I'm not sure what's the level in India of TB, but when I was working in South Korea, they was having a very strict rules for only not Europeans. They was not checking for TB compulsory. Rest of the world they was checking. In Zimbabwe, more than half of the population is sick, not caring, having a medical problems. And imagine if you have the bacteria that can reduce, not kill, just reduce the Mycobacterium tuberculosis. You already have something that helps. Again, we don't want to say that the bactericin will save the world. This is not a new antibiotic. It's not a completely things that will replace the antibiotic, but they can help to reduce. And another question, Santosh told me that it's about 50% subscribe for this course. Okay, let's do the calculation easily, 60. 1/3, one of every 3 persons actually carrying Mycobacterium tuberculosis in the body. This bacteria maybe will never develop, never. Will stay sleeping in your body for the rest of your life, but it's sky high. It means in this place about 20 people most probably having a Mycobacterium tuberculosis inside the body. Okay, don't be panicked. This bacteria will sleep there for the rest of your life, but it's a kind of common. And if you can have it again, probiotic or with bacteriogenic property that can inhibit Mycobacterium tuberculosis. So even Mycobacterium bovis or Mycobacterium avis, because they can be transmitted to the humans, it's already some positive things. Inhibition against some fungus. We're having this work showing that some of the bacteriocins can work against fungus. You see, already putting away of this whole definition, that's only closely related. You can inhibit some more than just a closely related. You just need to have a propertive model. You need to have very often using strains that are not pathogenic as a model in the lab, because we need to respect the safety in the lab as well. And then you can explore this specific interaction and actually develop new pharmaceutical products for the application in the human veterinary or agricultural applications. Synergistic interaction. Bacteriocins who work together with antibiotics, they can help each other, and in this way you can reduce the level of the use of the antibiotics. Anti-candida effect as well was reported. And Candida albicans could be a very serious vaginal infection. And treatment, in fact, with some bacteriocins and probiotics producing these bacteriocins could be a good option. Antivirus activity, I already mentioned, polio, herpes, measles virus can be treated. We have 99% reduction of herpes virus, 50% reduction of polio virus. It's not a medicine. Again, please don't ignore the vaccine, especially when you have the vaccine, but this additional treatment never, it's need to be ignored. Even control of cancer cells. There was a very good review published by 2 scientists from India, Kaur and Kaur. Must be wife and husband or brother and brother, I don't know. But they was really pointing this on this interesting effect of some bacteriocins. And interestingly, they can attack only the cancer cell, but they don't attack the healthy cells. We was working in Brazil on some project, and we was needed to check the cytotoxicity of some of our bacteriocins, and we used the cancer cells because it's much more easy to work with them in the lab, right? Who work with the cells? No one?
Cell lines, we don't have. Few labs they have cell lines.
Yeah, but it's, I mean, when we work with the cell lines, it's much more easy. The cancer cell lines, they grow easily, they develop easily, they don't want to die. Much more easy. Healthy cells, it's more difficult. But interesting, we have a very high cytotoxicity against cancers of breast cancer, cells of breast cancer. And it was like, okay, they are cytotoxic. But then a colleague of mine have it, okay, let's try with the healthy cells. No cytotoxicity. And then we repeat it again and again. very high activity against cancer cells of breast cancer, and almost no cytotoxicity against healthy cells. And then starting digging the literature, then was a report here, another one, then come this review from this scientist from India. There was summarizing all these things, and now it's getting a trend in the research because some of the bacteriocins have the specificity of the interaction with the cell of cancer, because the cancer cell, they have a different charge on the surface, they have some modification in their surface, and the bacteriocins can easily recognize them and actually kill them. And now even signaling molecule and quorum sensing, they're involved in all these processes. So the timeline, more or less, again, 95 polycines, first from the lactic acid bacteria, 28, 33 niacin was discovered, when authorized in almost around every country in the world. Received his name in 47. Why is such a big difference in the time? Why needed 40 years to receive the name? What's your opinion? Why? What happened in this period? Second World War. Do you think people were thinking about the niacin? They look how to survive at that time. So this was a reason to take a little bit longer. Well, authorized in Europe, in England, in the World Health Organization, US, etc. And then coming for this variety of applications again. Antimicrobial produced by lactic acid bacteria. You can have antimycobacterium. It's a good thing to explore. Antiviral properties, then could be bacteriocin, could be protease inhibitors. Antifungal properties, very often associated with different acids, especially phenylactic acid could be very often responsible for this. Antibacterial classes, different classes, bacteriocins and different other molecules. But important thing, if you look here, when you have the small amount of the bacteriocins, normally small amount is responsible for interspecies communication. This could be quorum sensing, could be quorum catching, or even between interspecies communication, when it's a small amount. Look like in the nature, bacteriocins being developed by the okay, sometimes I'm using this example. People that are very religious, okay, the God created bacteriocin. People that are very anti-religious, the nature created evolutionary process. How do you want, you can do it in your way. But important thing, when normally they're being created to use this interbacterial communication, they was not created to kill other microorganisms. They was much more important in this communication area between different microorganisms. Lately, when they are having in the highest level of the production, they can have bacteriostatic, bactericidal, or bacteriolytic effect. And when it's a little bit higher concentration, then you can have anti-cancer effect, they can have eukaryotic viruses, inhibition, or even effect against viability of spermatozoids. So it's getting more and more complex, but again, in the beginning we was exploring these antibacterial properties, but with the development of science we went to the communication and as well not typical killing properties of the bacteriocins. And develop of the history of the bacteriocins. This is taken from Scopus and from, let's say, how much this will be, last 80 years. When you put the term bacteriocin or bacteriocins, because very often in Scopus you need to put singular and plural to have better results, you see how exponentially growing. Well, it's even more because the data was being taken based up to the 24. But again, if you cross the word bacteriocin and antibiotics, or bacteriocin and cancer, here bacteriocin antibiotic, bacteriocin and cancer, you see as well develop of the research projects, publication of papers taking care for this. Well, here it's not too much, it's like 60-something papers per year, but it's already starting because it's showing a potential of these products against cancer cells. How much time I have? What time I need to finish?
We have lunch time and another 15 minutes.
Okay, at 1 o'clock I need to finish. Okay, so we can talk about mode of action, and tomorrow we continue. By the way, in the afternoon, Santosh promised you to try to isolate your own probiotic. It's exciting because you will find that in 1 week you have something in your hands that giving a potential. But then you need to study on this, and we spent like 2-3 years to show what you have exactly. Okay, how they work. Most of the bacteriocins, actually they're using, associated with the proton motive force. And they can make a pores inside the cell wall. And in this way, they can actually interact with the protein motive force. This is actually its potential energy generated by the electrochemical gradient established between the sides of the membrane. But if you make a pore inside the membrane, obviously you cannot anymore control this balance. And so in some cases, this will interact with anchoric molecules, and they facilitate the interaction of antimicrobial peptides in general, bacteriocin more specific, with the plasma membrane. This actually increasing problem with the stability, and you have evolved lipid 2 and system mannose phosphotransferase system. So lipid 2 and mannose phosphotransferase system, they actually are very important because they can serve as a receptor for some of the bacteriocins, and different transporters as well can be involved in this process. It's interesting that some of, okay, do you know how works the venom of the cobra? Do you know how it works? They block some channels on and they interrupt the transferring of the signal, they interrupting with the stability of the cells. Same thing. If you use the bacteriocin, they can interact with these channels, specific channels on the cell surface, and in this way they can actually interfere with the cells on the integrity. In the laboratory, sometimes using the poisons from the scorpions, from the different snakes, including this one that is like naja. Yeah. Using the poisons for them, because in this way we modulate the different channels in the cells, and we can see if the bacteriocin attacking the same channel or not. And we can actually make a conclusion how the bacteriocin actually killing the pathogens. I don't want to see the snake. Sorry, I had the phobia. If I see the snake here, you see me in the airport in the 10 minutes. I will run to the Delhi International in 10 minutes. Bacon will be the new Olympic champion from this university. But the important thing is formation of this interaction. And very important role is here of the lipid 2, because remember from the antibiotic courses, most of the several antibiotics, vancomycin, etc., they interact with the lipid 2 in order to make a contact and to disturb the cell membrane. And lipid 2 is actually in the phospholipid layer, and translocation of the periplastic space to the membrane of murin synthesis, okay, it's a classic microbiology of importance of this lipid. But again, bacteriocin can interfere with the lipid 2. And since the lipid 2 is important for the cell, you cannot take it away. If the cell losing lipid 2, we'll have the problem with the murin synthesis. So, what's happened? Bacteriocin cannot develop resistance, because the receptor is there. You cannot take away the receptor. If the bacteria losing the receptor lipid 2, she will die. But then, bacteriocin is coming and recognize the lipid 2 and kill. Well, for now, we don't have such a strong resistance to bacteriocin. For now. But remember when Fleming developed the penicillin, well, everyone was happy. We finished with the disease, no any more disease. Well, 10 years later, problem with antibiotic resistance already started. Same thing, for now, we are happy. Sometimes I want to say, the development of new antibiotic, development of new medicine is a kind of battle. We win the battle, but we don't win the war. War is still with the pathogens, is in the front of us, and will be, because obviously microorganisms are not stupid. They will develop some kind of resistance. They don't want to be just stand there, okay, kill me. I know you don't like me, kill me. No, they will try, they will find a way to protect themselves. And maybe in 50 years we will have the resistance to bacteriocins, but at least now they work, and we need to explore this. So Murin side chain and cross links. Murin, the final product of the lipid 2, is interacted by the complex network of amino acids chain, so everything's okay. And this is the point that the bacteriocin come and can interact. So they can be bacteriolysins, or they can be bacteriostatic. Well, this is a different story, because sometimes we don't kill them, but we push them down, and then it's coming the different mechanism of the host that can influence. It's not always that we can have the bacteria and they will solve all the problems. We need to have cooperation with the other systems of the organisms and the way to protect from the pathogens. Well, some of the bacteriocins like enterocin, enterolysine A, they can interfere, they can go inside the cell wall disruption because they can incorporate in the phospholipids. But if you destroy the phospholipids, you are actually making the cells more easily to be targeted by antibiotics, they more easily to be targeted by, even by the acids. So again, not a single molecule, it's a complex of different molecules. In fact, it's the way to work better. Mannose phosphortransferase system, PTS, is associated with the spontaneous complexes, and when you interfere with this system, you block the system or you open it completely, you interfere with the metabolism of the cell, and it's work. Then you can have the bacteria, especially if you're gram-negative, they can interfere with the RNA polymerase. They make a simple complex with the RNA polymerase, but once this protein, bacteriocin, make a complex with another protein, with the polymerase enzyme, well, polymerase is not working anymore. You block the RNA polymerase, what will happen? Not going to nothing good. Or DNA GRAS, again, one enzyme combined with another protein, two proteins making a complex. Complex, it's interrupting. It's like an enzyme inhibitor. They work in a very simple way.
So what is the concentration at which they work?
Well, this is an interesting point. They work very well even in nanomolar, very small amount.
So when you were testing it against the cancer lines, what concentration worked?
Well, the question is, when we work with the bacteriocin, we don't checking their concentration as a milligram protein or nanogram, whatever. We're checking them as arbitrary units, like enzymes.
So was it a pure preparation?
So normally we're using our own preparations, normally purify them, very often as a semi-purified, it's much more easier and cheaper. And you don't need the equipment. You can do it with a very small column and syringe in the lab. Only you need to put 5 students because you need to do drop by drop, drop by drop. But it's cheaper than by the HPLC. Well, in one moment we need to go to the pure material. But normally even in the 50 arbitrary units per ml, they're having a good effect. Very often this is corresponding to some microgram of protein, enough. Bacteriocins can be combined with antibiotics again, because they work in the similar way, but they're having a different targets. And if you target pathogen on one side, in one receptor, you disturb the membrane, and then coming antibiotics, they're doing a completely different mode of action, obviously will work even better. So vancomycin, niacin, and nisin, they work together. There's several work in the treatment of methicillin-resistant Staphylococcus aureus. We're having some microcins and chloramphenicol. There was as well some gram-negative bacteria can be worked. And in general, when we talk about their mode of action, we need to keep in mind the 2 principal ways. First way, the bacteriocin is coming, there is a lipid 2, N-terminal of the bacteriocin will recognize the lipid 2 because they can make a recognition, and the C-terminal will go inside. And it's interesting that most of the bacteriocin that have an anti-listeria effect, they have a very constant N-terminal part of the molecule. And they will always recognize lipid 2. This is why listeria is very easily to kill by some bacteriaceans. Some of other, like class 2 saccharins, they going and by electrochemical interaction, they will interfere, they will incorporate as a protein inside the membrane. Remember the membrane of gram-positive, gram-negative, they have proteins in there. And they have this kind of mimicking, like, I'm your protein, I'm protein from your membrane, and they can come and incorporate. Or another one, that is one that one mentioned, that interfere with the phospholipids, bacteriolysins. They go, they make this complex with the phospholipids, and they destroy completely cell wall. Again, too much work is done, but we're still on the top of this iceberg. It's a lot to explore, especially in the mechanism of interaction, because, as I mentioned, sometimes a specific receptor in gram-negative could be responsible to have this contact with the bacteriocin, and bacteriocin from lactic acid bacteria can interact, and they recognize the receptor, they go in the membrane, and they destroy it. In Sao Paulo, if you leave your house with the open door, you come, you find nothing. It's quite sad, but crime existing. It's not too bad, but, well, exists. And the same thing, if you open the bacteria, you open the pore, you will lose everything, for sure. And different method, different receptor, interaction with RNA, with proteins, enzymatic proteins associated with the cell life of the cells could be a very good option to see how they work. And then, finishing really quickly in a few minutes. So now the bacteriocin is produced. And normally bacteriocin is produced, this is the bacteriocin, this is the leader peptide. It's a complex molecule, but in this way the molecule is not killing nothing, because it's two parts that are together. In this form it's not active. But when it's going via the ABC transporter, one part it's losing, and you have active bacteriocin. After that they will do work. But then, interestingly, these bacteriocins can actually interfere and can activate production of the immunity proteins, so that bacteria can protect themselves from the bacteriocin, because bacteria maybe have lipid 2 as well, and if have lipid 2, this bacteriocin maybe will try to kill his own producer. Well, it's not good. So, but then we have the activation of the immunity protein because of this interaction. Then some of the bacteriocins can go to the neighboring cells and recognize this histidine protein kinase. And recognizing, we're activating this losing the phosphor from ATP, DTP, dephosphorylation? Well, losing phosphor from ATP to DTP, and this phosphor, in fact, will be serving as a responsible regulator for activation of the gene on the DNA level to start producing activating all the operon responsible for the production of the bacteriocins. So one bacteria producing the bacteriocin go to another bacteria from the same, his brother, sister, whatever how we want to call it. And by this system, we will activate bacteriocin production in the next one. Another interesting point, some of these inductors actually coming from the pathogen. Listeria have the surface proteins that are able to be recognized as a pseudobacteriocin by the cells, and will start inducing production of the bacteriocin. So, okay, look guys, we're having a listeria around. We need to produce our bacteriocin to protect against listeria. And this circle is actually going and going and going. But then some of them, bacteriocin, they have this leader peptide. This is the bacteriocin leader peptide. And when it's going via the ABC transporter, they need to cut and liberate the real bacteriocins. But some of them, they are leaderless. They don't have leader peptide. They are produced, but then this interesting point, on the N-terminal, they always have, methylated methionine. And this fact that have this methylated methionine protect the own producer by effect of his own bacteriocins. Let me just quickly, I can finish in 5 minutes. Okay, Now, you have it this schematic way. So you can, at the top you cannot see it, but this is the same bacteriocin there, and they have this leader peptide in the front. They need to be liberated. After that, the bacteriocin very often can make these disulfide bridges between some non-specific amino acids. You can have methylamine, lanthanoine and beta-methyl-lanthanoine, not proteic amino acids. But you can make these disulfide bridges that will give a specific structure. But then you have this cleavage that will be done here. Leader peptide will be liberated when going via the ABC transporter. And when it's going out, you're going to have only this part. In case of the lysine, lactocin, another lactocin, but going out already with these disulfide bridges. But this structure actually is important because only in this case, the molecular of the bacteriocin can have the specific configuration to recognize the receptor. If it's not this structure, no recognition, no effect. Then it's by the way, I'm going to provide you these slides. Santosh, we can give it to them, right? You need to decide how we're going to distribute the slides. You have in here, it's like a story. Again, you have enterosin TT, various genes associated, various product of the gene expression. But anyway, it's important to see that you have a activation of the specific protein. Its fact is coming to enterosin F, that is induction factor. This induction factor needs to make contact with enterosin K, this kinase enzyme. They will activate enterosin R, that is a regulation factor. And only in this case, expression of the entire operon will be available. So no choice, you need all these steps in order to produce and bond. Classification of the bacteriocins could be very different. In the past, we have several classifications. Very often, people just put a new classification to have more citation. Well, it's okay. But most of them, they say antibiotic class 1. It's a classical one. They are smaller than 5 kilodalton. They have lanthanoine and beta-methyl-lanthanoine and some unusual amino acids. They need to make disulfide bridges in order to have the active molecules working. There are several of them. They are cationic normally, negative charge. Some of them could be positive charge, which majority is negative. Some of them could be even globular. Then you have class 2, the smaller than 10 kilodalton. So there is a nightmare sometime in the protein work because small protein is more difficult to work from some point. But the good point, they're thermostable because they're small, they have nothing to lose in the high temperature. Very important part is 2A pediosin-like bacteriocin. They have this activity against listeria, and they have always this conservative amino acid. So you can easily target this part of the molecule on the genetic, on the protein level. You can easily discover if they have this fragment and on the N-terminal. There is some bacteriocin, they're a nightmare for the work because there are two components, two proteins, and they need to be together to work. If you separate, they don't work. And when you purify them, how you can reconstruct them? It's difficult, but it's fun to work with them. Again, this sec pathway, this one they don't have this leader peptide as well. And worst scenario, if you work with the bacteriocin, you have the bacteriocin that is a complex of protein and non-protein molecule. Because you need to have a protein on one side, you need to have carbohydrate and lipid to get together in order to work. You do the protein purification, you lose the protein, lipid. You do the protein purification, you lose the carbohydrate, and then it's not working. So how you know that you purify? But again, it's more fun to work. Then it's a classification from Paul Potter. He just said, very simple, class 1 antibiotics, class 2 non-antibiotic, very simple, but it's a kind of difficult. And I like much more this of John Tack from New Zealand, because it's again, antibiotics, they can be linear, they can be globular. Niacin is coming here in this group. Class 2 small non-modified peptide, because they have this small modification, this disulfide bridges that will appear and non-specific amino acids. And here class 2, they are non-modification, pediosin-like. You have class 4 that is cyclic peptides because none of them are linear, some of them could be cyclic. And you have class 3, very big proteins that have this antimicrobial activity. Okay, let's stop here for this morning, and tomorrow we continue, actually not tomorrow, afternoon we continue with some practical. And when we do the practical, actually we can see why all this information is actually linked to the manipulation. Questions can be in the after lunch?
Yes.
Okay.
Thank you very much, Professor W. This was the afternoon session. We will be having more interactive sessions. Welcome the Department of Genetics seminar. He will interact with each of the participants, their interest, how to avoid hands-on training, how we can isolate these probiotics, how we can grow it. So we'll have more there. Go for have a quick break after the lunch we meet in the Department of Genetics. Thank you very much for your service.